Hi there. I'm going to do a quick video here on an AS topic, AS transport in plants. Um, to get most benefit from this video, you'll need access to a set of my notes because I'm constantly referring to the notes and to pages and to certain sections of the notes. And I'm going to use the notes as a guide to ensure that I don't leave any parts out of this section. You'll also need access to a set of questions. I'm going to try and incorporate the questions into the video. So as I do each section in the topic, I'm going to try and go through the questions fairly quickly. I'm going to move fairly quickly. You should also, if you have the notes and the questions, you'll also have the answers. So you can also have the answers in front of you there and compare the answers with how I describe the answer, if it gives you more clarification. Okay, so I'm going to, as I say, I'm going to move fairly quickly. It's quite a long topic, quite a difficult topic for AS students. Okay, so here goes then. Right, this topic is about plant transport. Essentially, plants, inside plants you have transport systems that transport water and ions. That would be the Xylem Vessels, and a transport system that transports the products of photosynthesis, which would be the Floam Vessels. Xylem Vessels and Floam Vessels, and you may have come across those to some extent at GCSE. So at the very start of the topic, you're looking at the structure of the root and the stem and the leaf, and identifying where these different tissues are at. In other words, where are the Xylem vessels located in the root, where are the Floam vessels located in the root system, and so on. As you move up the stem, then you're looking at the location of the Xylem vessels in the stem and the Floam vessels in the stem. And eventually you get into the leaf then, and some discussion about where everything is in the leaf. So I'll leave you to essentially learn the diagrams yourself and look at the diagrams. And I'm going to move quite quickly then to look at uptake of the root system. In other words, how the root system of a plant takes up water and dissolved ions from the soil. Now, ions are often referred to in biology as minerals or elements even. But I suppose the best answer is, the best description is ions, because what the plant takes up from the soil essentially is water with ions dissolved in it. So the water that comes up through the root system contains magnesium ions and calcium ions and potassium ions and so on and so forth. And at GCSE you would have learned a wee bit about what each of those different kinds of ions, what the plant needs with those ions. GCSE might have learned that the plant takes up nitrate ions dissolved in the water, and nitrate ions are needed to make proteins for growth. Magnesium ions are needed to make chlorophyll. Magnesium is an atom in the chlorophyll molecule. Magnesium and calcium ions were needed to make calcium pectate, which is part of the cell walls of plant cells. So those are just a few of the different ions that are dissolved in the water that's taken up from the soil and taken into the root system. Now that water then makes its way eventually into Xylem vessels, into Xylem vessels, because Xylem vessels are the pipes or vessels that are going to carry this water with dissolved ions around the plant. So we start off first of all down at the root system then, and look at the location of the Xylem vessels in the root system. And you discover when you look at a cross section of the root system that the Xylem vessels are located at the very center of the root. The root is sort of cylindrical in shape, like the shape of a carrot. I sort of imagine a carrot as a root of a carrot plant. And the Xylem vessels are in at the very center, and they're arranged in a sort of star-like formation of Xylem vessels. So the first element that we have to look at is, how does the water and dissolved ions that are absorbed at the surface of the root system, how they make their way across the root system into the Xylem vessels, which are at the center. Now, GCSE might have learned that ions are absorbed by root hair cells, And root hair cells are at the very surface of the root system. Actually, they're in a layer called the epidermal layer. So you need to know your layers of your root system. So the outer layer is called the epidermis, epidermal layer, and through it you get lots of root hair cells. With root hair cells are simply cells with adaptations to create a large surface area for uptake of water and ions. And then inside the epidermis layer, with the outer layer, then you have a layer which essentially almost runs to the center. It's a layer of cortex cells, which are big round cells. And then towards the center, you come across another ring of cells, or a layer of cells, called the endodermis. And it's inside that ring of cells, the endodermis, then that you have the Xylem vessels. So the first kind of question that sometimes comes up on this topic is a discussion of how water and dissolved ions make their way across the root system from the outer epidermal layer right across through the cortex cells right the way into the very center of the root system, where they then can enter the Xylem vessels, which then carries the water and ions up the plant. And here you're introduced to this idea of apoplast and subplast pathways. So the places that I'm looking at here, I'm looking at page 2 in the notes there, because on page 2 I have a diagram showing a cross-section of the root system, and you can see the cells here in the middle. These cells are called cortex cells, and it's across those cortex cells that water moves, water and ions move. And water and ions move across those cells by apoplast and subplast pathways. And here I'm going to refer to page 3 in the notes, and I'm also going to look then, I'm going to refer you to a couple of questions. Question 1 and question 3 in the booklet. So question 1's a recent question about this, and question 3 is an older question. I like question 3. You can look at question 3 first of all. Question 3 is quite easy to understand, because in question 3 you've got the root hair cell out at the edge here, and then you've got these cortex cells running across, and then you've got this layer of cells called the endodermal layer. And it's asking you to identify the cells. B would be cortex cells, and A would be endodermal cells or endodermis cells. And you have to add hours then to show the apoplast and subplast pathway. That's essentially pretty straightforward. I'm referring you there to page 3 in the notes. Essentially, you have to draw arrows to show the movement of water across these cells. So water basically has to move from the root hair cell through these layer of cortex cells to reaches the endodermis and so on. And it moves by 2 pathways, the apoplast and subplast pathway. Now, apoplast, first of all, apoplast pathway simply means that the water enters the cell wall of the root hair cells. Now, remember, the cell wall is on the outside of the cell, and then inside it you have the cell membrane. In this case, water, we can consider that water just goes into the cell wall, and then it moves from cell to cell by simply being sort of soaked. It's not the right word, but it moves from cell to cell by moving from cell wall to cell wall. So it's as if water just stays to the cell wall, never goes in through the cell membrane. And so you're sort of drawing a line that runs the water the whole way from cell wall to cell wall until it gets to the endodermal layer. And then very importantly, at the endodermal layer, you have to show that it can't get through this impervious layer or impermeable layer. There's a layer in the endodermal layer called the Casparian strip. So at that point you have to show that the water can't get through that, and the water has to exit the cell wall. That's supposed to be the cell wall, and it goes into the cytoplasm of the cell, and then it moves through these what are called Plasmodesmata. So that's the apoplast pathway. Water moving from cell to cell across the cortex cells from through the cellulose, through the cell wall. So I imagine it soaking through the cell walls. And then the subplast pathway is maybe more obvious. Water enters the root hair cell, and then it moves from the root hair cell to the next cell through a little channel that goes between the cells. And these little channels are called plasmodesmata, and they're basically like little channels of cytoplasm between plant cells. So in other words, if you were in one plant cell and there's another plant cell next door to it, you might find there's a hole in the wall. If you were in the cytoplasm of one plant cell, you could actually make your way through this little channel of cytoplasm that goes to the next cell, and that's called a plasmodesmata channel. And these plasmodesmata channels then between plant cells allow the transport, for example, of water and ions So essentially, you go into the root hair cell, then you go through that little Plasmodesmata channel, and then you keep going through the little Plasmodesmata channels and make your way the whole way across until you get to the endodermal cell, and then you can make your way into the xylem vessel that way. So those are apoplasms and plas pathways, and you can look at those, look at that particular question and answer that question. It says here, part 5 of that question says, what makes up the Casparian strip? Casparian strip is made of something that's impermeable to water, or doesn't let water through, and it's called suberin, S-U-B-E-R-I-N. And basically its purpose is it asking that question, what's the function of that suberin in the Casparian strip? And the answer is to prevent the water continuing by the apoplast pathway or through the cell walls and forcing it to go in through the cell membrane and go by the symplast pathway. Forces it to go leave the apoplast pathway and go by the symplast pathway. That's the answer to that question 3 at the bottom. Now, question 1's a more recent question, and it says, essentially, it's the same sort of question. Use the diagram and I'll explain. This is part 3. Explain fully the function of the Casparian strip. So once again, you would say it stops water moving to the apoplast pathway and forces it to go by the symplast pathway. It forces it in through the cell membrane. Forces it in through the cell membrane of the endodermal cells into the cytoplasm. And it says, what's the function of it? And the answer that they allowed here was to bring the movement of water and ions under metabolic control. Bring it under metabolic control. So I wanted then just to say a wee bit more what that means. What do we mean by metabolic control? So it said on page 4 there, again, we sort of explanation of that, but I don't want you to dwell on this because there never has been an extensive question on metabolic control. It's simply just to understand that it exists, metabolic control. So what's meant by metabolic control? And essentially the answer is this, that if the water and ions have to move from the cell wall, if they have to go in through the cell membrane to get into the cytoplasm, then as the ions move through, as the ions approach the cell membrane, they get involved in active transport. There are active transport proteins in the cell membrane that lift the ions from outside the cell membrane and move them to the inside. So you would say the ions that are present in the water are actively transported into the cytoplasm by active transport proteins. Those active transport that process of active transport involves energy, and that's where the metabolic aspect of it comes from. The idea that the cells have to provide energy for this active transport, and by moving the ions into the cytoplasm, then that creates a high concentration of ions in the cytoplasm, which lowers the water potential of the cytoplasm, which causes the water that was moving with it, causes the water to follow after the ions into the cytoplasm. So the water then moves down a water potential gradient. So in short, by moving the ions across the cell membrane, you bring them under metabolic control. What does that mean? It means the ions are moved across the cell membrane by active transport. And when the ions are moved across by active transport, that creates a high concentration of ions in the cytoplasm, i.e. a low water potential. Then that causes water to follow after the ions across the cell membrane down the water potential gradient, and that essentially is metabolic control. As I say, it's never been asked about, but essentially that's what it is. Okay. So that's questions 1 and questions 2 there, which are related to that area. Now, I want to move then quickly on to a discussion of the xylem vessels. So essentially then the xylem vessels are the pipes vessels that are going to carry the water and ions up the stem of the plant, up to the top of the plant, up to the leaves, and up to the top of the plant. And you often get questions asking you about these xylem vessels. And the key thing about these xylem vessels is they're little pipes carrying water up. The key thing about them, is often asked about them, is that they are strengthened or stiffened by a material called lignin, which is a kind of a material that's put in the walls of the xylem vessels to make them stronger, make them stiffer and more less likely to collapse as the water flows through. And now, in a moment or two, we'll discuss why it's important that the xylem vessels don't collapse. But just let's go with the idea, first of all, that the walls are stiffened by this stuff called lignin. Now, the arrangement and the common question that gets asked about this, I'm looking here at question 7. Question 7 is a good question to look at in the question booklet. It's on page 8. and it's asking you about 7 part C. And it shows you the Xylem vessels. The Xylem vessels have lignin in their walls, and the lignin is arranged in different patterns. The lignin can be just like rings of lignin around, and that's referred to as annular. So in question 7 part C, there above annular, if you write rings of lignin, or it can be in like a spiral pattern. Lignin can be in a spiral pattern. So you would rings or spiral. And what I often do with students is I say, draw a line. I want you to draw a line after the spiral and before the scalariform. So just draw a line straight down the page there. And on the left of that line, I want you to write a young plants. So young plants over here on the left. And over to the right, I want you to write older plants. Young plants and older plants. And essentially, here's the idea. You got all these different patterns, ring pattern or annular, it's called, or spiral, or all these other scalariform, reticulate, and pitted. By the way, you don't need to know those words, scalariform, reticulate, and pitted. Those are not important words. They're not asking those words. But be aware of these, the idea of annular or ring pattern or a spiral pattern. And here's the story. Younger plants. If a plant's quite young, and you look at the Xylem vessels running up its stem, you'll find these kinds of patterns. So you find the spiral and annular pattern in young plants. Whereas older plants have these other patterns. And the question, so first of all, leave part 1 for a second or two. Part 2, suggest why the Xylem vessels in young plants have rings or spirals. And here's the answer. Rings or spiral patterns allow the Xylem vessels to extend as the young plant grows. So rings and spiral patterns allow the plant to continue to grow. The Xylem vessel can grow and the spiral simply stretches out, or the rings get further apart. Whereas those other patterns, scalariform, reticulate, and pitted, those are fine plants that are much older. They've stopped growing. Their Xylem vessels won't be getting any longer, so they don't need to stretch. So essentially, that's the answer to that. And you see that's indicated at the bottom, the notes at the bottom of page 5. Young plants have annular or spiral patterns. These patterns allow the Xylem vessels to get longer as the young plant grows in length. Okay. That question has been asked recently too. I'm just looking here at question 10C in the booklet there. And you'll see it shows a kind of a photograph. It's a very difficult photograph to see, but it's supposed to be of a Xylem vessel. So that's the Xylem vessel there. And that's the pattern of lignin. And you can see it looks like a kind of a spiral pattern. And then it says, down part 2, identify evidence from the picture that this is young Xylem vessels, and explain the advantage. So you say, oh, it's young because look, you've got spiral patterns, and that's typical of young plants. And then explain the advantage of this, and you'd say, young plants are continuing to grow. Xylem vessels are stretching, getting longer, and the spiral pattern will stretch, will get longer. It'll allow the Xylem vessels to get longer. And so that's essentially that. Now, in a moment or two, I want to say about why do we need this strengthening at all in the walls of the Xylem vessels. Well, maybe I just deal with it now. Essentially, it's kind of dealing with how water moves up the stem. How is water moved from the root up to the top of the plant, up to the leaves? And the answer is essentially that the water is sucked up. It's sucked up. Now, in the exam, you wouldn't say that. But basically, the idea is the top of the plant, the canopy of the plant, the leaves up at the top of the plant are sucking the water, and whatever's dissolved in the water up the plant, it's been sucked up. Now, in the exam, you don't say that. You'd say the water's drawn up by negative pressure. You can say by suction forces, possibly. But the correct description of these forces are cohesion tension forces. Cohesion tension forces are involved in moving the water up the plant. And because the water and that dissolved ions are being sucked up through the Xylem vessels, that causes the Xylem vessels to collapse inwards. And this would be evident to you. You would maybe understand this if you had like a straw and you put a straw, say, into some water or some juice or drink or whatever, and then you sucked through the straw. What you would find is that the straw is liable to collapse inwards. it's liable to collapse. And this is very evident if you have a paper straw. Paper straws aren't common nowadays, maybe they are, but paper straws used to get really wet, and then as soon as you were sucking through them, the paper straw collapsed inwards. In other words, if you're getting a suction effect from the top, the Xylem vessels are the forces referred to as a tension force. It would cause the Xylem vessels to collapse inwards, not to burst outward, but to collapse inwards. And therefore, that's the purpose of the lignin. And in those two questions where it asked there, in question, sorry, just going back then to question, get that question again, question 7. 7 part C, where it says, explain why the added strength, this is 7C part 1, why is the added strength of lignin needed in their function? And the answer is to prevent the Xylem vessels, to strengthen the Xylem vessels, to prevent them collapsing inwards under the suction or cohesion tension forces which are drawing water and ions up the plant. So that's that question. And then that same thing's also asked in question 10C. They've asked the same thing. It's 10C. They're saying, explain how the strong forces arise within the xylem and describe how it's adapted to withstand these forces. You'd say the strong forces are due to the suction or cohesion tension forces which are drawing water up through the xylem vessels. And how is this? How is the xylem vessel adapted? You would say it's got lignin. Lignin deposited in its walls to prevent the xylem vessels collapsing inwards. We'll talk a wee bit more about that as we go on. So that's it. Now, turning then on to the next section, which gets asked about 2. On the next, an E, section E there on page 6 of the notes, you've got a cross section of the stem, and there you're made aware that the xylem vessels in the stem aren't arranged at the center of the stem, but they're arranged in groups. The xylem vessels are arranged, so if that's the cross section of the stem, the xylem vessels and the floam vessels are arranged in these things called vascular bundles, which are arranged out round the edge of the stem. And each vascular bundle has arranged that, and the xylem is located towards the center. X, and just put X for the xylem. X is there. And then the floam vessels, they're out here. Floam's out on the outside. So the xylem vessels are towards the center, and the floam vessels. So basically, there's like a ring of vascular bundles. They're called vascular bundles, and they're arranged around the stem. Now, next thing we're doing is basically addressing in more detail how exactly is water moved up the plant from the roots up to the top of the plant, because it has to. It's going against gravity. The water's got a weight. It has to either be pushed up or pulled up. Essentially, it could be pushed up or pulled up, or maybe a combination of both. Well, actually, if we look into it clearly, I said, how is water moved up the stem? Actually, there's sort of, it's both pushed and pulled. But the pushing is pretty irrelevant. The pushing is created, remember I said that at the root system, you're having active transport taking place, and ions being pumped across into the endodermal cells. Well, that causes water to move by osmosis into those endodermal cells, and you get this buildup of water down at the root system, and that's often referred to as root pressure. And that buildup of water, and that is, to some extent, that pushes water up the stem, but it's very minimal. I would say it's probably less than 10% of the effect. 10%, it provides less than 10% of the effort needed to get the water up the stem. By far, the vast majority of the effort to get the water up the stem is a suction thing. It's a suction, and as I've said, the suction due to the leaves. the water is actually pulled up. So this idea that when you look at plants are actually pulling water up to the top, by the leaves are sucking the water up. So I don't like using the word suction too much, but basically it's got a correct word. It's called cohesion tension forces. And then you have a theory called the cohesion tension theory. You could call it the leaf suck theory, but that wouldn't sound maybe as good. It's called the cohesion tension theory. And that basically explains this mechanism by which water is drawn up or sucked up to the top of the plant. And I'm just looking here, I'm looking at page 7 in the notes, cohesion tension theory, and I'm just going to refer to question 6 in the question booklet there. Question 6 is probably the best illustration of this. It doesn't ask a direct question about the cohesion tension theory, but it's got a good diagram. And now question 6, you'll see question 6 has this diagram, and you'll see I have the same diagram in the notes there in quite a small fashion. And essentially, this diagram is it's a cross section of a leaf. And so cross section of a leaf. I suppose I should do a leaf in green here. And so that'll be the top, and that's the top of the leaf, the upper epidermis, and then you have palisade cells, spongy mesophyll cells, and so on. So you have upper epidermis, and then you have tall palisade cells like that. And then you have spongy mesophyll cells, with a few more palisade cells, and down to that. And then you have the lower epidermal cells like that. Upper epidermal cells, I just put a few of those. Palisades, spongy mesophylls, and lower epidermis. And then, of course, you have guard cells here, and guard cells, as it were, guard the stomata. So there's lots of spongy mesophyll cells in here now. More spongy mesophyll cells. Okay, so this is spongy mesophyll cells. So essentially, and of course, in here you'll also have your veins. Veins in a leaf. So if you draw a leaf, if you're just sketching a picture of a leaf, often you do a leaf like that there just to show. And these here would indicate, these would be like, you call these the veins of the leaf. Now, if you were to dig down into those veins, you discover that that's where the xylem vessels are, because the xylem vessels are running through the leaf in like a spider pattern. And so if we were to put some in the cross section here, put some xylem vessels, and that's just and put some xylem vessels there in that region. Yep. So essentially, how we go about talking about the cohesion tension theory is as follows. Let's get rid of that cohesion tension theory. So rather a big stomata there, but that'll work okay. So this is our stomata here. These are spongy mesophyll cells. Now, I'm just going to go fairly quickly through this. Essentially, we're asking the question, how does a leaf suck water up the stem, up to the top of the plant? And so you're going about this thing, you say, oh, this is the basis of the so-called cohesion tension theory. And here's how you start. And I'm using this is a good diagram on question 6 there. Essentially what you do is you say this. You say that water that's inside the leaf evaporates off the surface of the spongy mesophyll cells. So there's water evaporating off the surface of the spongy mesophyll cells and evaporates into this airspace. So this airspace becomes full of like water vapor. And then you say this, once the water is in here, you get a high concentration of water, and then the water diffuses out through the stomata. Out through the stomata to the outside. So that's the first 2 points here, that the water is evaporating off the surface of spongy mesophyll cells into the airspace, and then it's diffusing out through the stomata. And those 2 points make up a description of what we mean by transpiration. So later on you'll see a question where to ask, what do you understand by the term transpiration in plants? And if you get that question, you have to say these 2 things. You have to say, transpiration involves the evaporation of water off the surface of the spongy mesophyll cells inside the leaf into the air spaces, followed by the diffusion of water out through stomata. And that's your definition of transpiration, or stomatal transpiration as it's called. Okay. And so that takes up the first 2 points there. Now, if water is evaporating out of these cells, these spongy mesophyll cells, out to the outside, well, that water has to be replaced from somewhere. And the answer is that the water is replaced from the xylem vessels, which are bringing water up to the leaf. So basically, then you'd say, water that evaporates into the spongy mesophyll cells is replaced by water from xylem vessels, which are present inside the leaf. And that's point number 4. Now you get a mark then for saying that the water can travel through the spongy mesophyll cells inside the leaf by apoplast and symplast pathways. In other words, the water can go from this cell to this cell to the next cell by apoplast and symplast pathways, just the same way as it can travel through the cortex cells in the root system. So there's a mark for saying that. And you get a mark for saying, for mentioning that water molecules, as they travel by apoplast or symplast pathways, they are attracted to each other. They're held together by a force, and this is an attractive force between water molecules. This is a bit of chemistry or physics, I suppose. Even water molecules are attracted to each other by forces. It's hydrogen bonding, it's called, but it's also referred to as cohesive forces. There are cohesive forces between the water molecules. And now, the thing about these cohesive forces is this, that you can argue that if you follow some water molecules through these cells here and evaporating, so there's some water molecules traveling there through the cells and then evaporating here. You can say this, you can say something like, as a water molecule evaporates off the surface of a spongy mesophyll cell, it pulls the next molecule behind it along, as if the water molecules form a long chain of water molecules. So each water molecule that evaporates pulls the next water molecule on, and then it in turn is an attractive force called a cohesive force with the water molecule behind, such that you can kind of trace if you go right the way down through the plant and down the stem of the plant, right down to the root system, and you can say that there's like a long chain of water molecules extending the whole way from the leaf right the way down to the root system. When I say a chain of water molecules, I mean water molecules all sort of attached to each other or hanging on to each other by these cohesive forces, such that if one water molecule evaporates here, it pulls the next one behind it, and that in turn pulls all the water molecules the whole way up from the root system. And this is sometimes referred to as long unbroken chains of water molecules being pulled up through the plant, all held together by cohesive forces. Now, because the water molecules have been pulled up the stem of the plant, the water molecules are been held onto each other, but there's stretching forces because of the weight of the water molecules. If you're pulling a big long chain of water molecules up and they're all held to each other, there's a weight involved there, and so the long chain of water molecules is being stretched, and that's what we're seeing is happening. The water molecule, the water molecules that extend the whole way down the xylem vessels are under a suction or stretching force. So whenever the suction or the pull, whenever this loss of water molecules happens here, all the water molecules behind are the chain of water molecules has been stretched, and this is referred to as a cohesion. This is referred to as a tension force, a stretching or tension force. So I said that due to the width, the chain of water molecules are under a stretching or tension force. And that's where the lignin comes in there. The lignin, if you have a stretching or tension force in the water molecules as they move up through the xylem vessels, they'll cause the walls of the xylem vessels to collapse inwards, the suction force. And so this is where the xylem layer protects. So that's your cohesion tension question. Hasn't been asked in any great detail, like that, but it would serve an essay question. You can see it's 9 marks there, but 9 marks. You could part of an essay in answering that question. So over the page, then we introduced the idea of transpiration. And as I said, transpiration, if you get asked to define it, you say it's the evaporation of water molecules from the spongy mesophyll cells into the airspace, followed by their diffusion out through the stomata. So they've been lost out through the stomata. And when they've been lost, this loss of water molecules out through the stomata is called stomatal transpiration. Stomatal transpiration. So yes, there's other kind of transpiration. And the answer is there's a very small amount of transpiration through the waxy cuticle. But as you were told at GCSE, the waxy cuticle prevents water loss. Actually, it doesn't. It greatly reduces it, but it isn't absolutely zero. So some water is lost through the cuticle. And this is evident in some plants that have to try and reduce water loss, called Xerophytes, and you'll find that they often have a thicker waxy cuticle. That suggests a thin waxy cuticle might let water out. So a thicker waxy cuticle. So okay. So moving on then to this idea about transpiration. So transpiration essentially at GCSE was just the loss of water from plants. And you would have said the leaves were mostly responsible for loss of water from the plants. And if you're asked at GCSE, you'd say, oh, the water comes out through the stomata, and that's where it gets lost. Maybe more precisely described needs to talk about evaporation and diffusion. Now, the rate of transpiration is essentially the rate of loss of water from the leaves of the plants. Essentially, it's the leaves losing most of the water. Rate of transpiration, as I've said there on page 9, is affected by things to do with the plant factors, and also to do with the environment. And the relative question, the relevant question here is question 11. The essay question in the booklet here, question 11b, where it asks about factors that affect the rate of transpiration. The transpiration, the rate at which a plant is losing water to the environment, is affected by things to do with the plant and things to do with the environment. At GCSE, you would have said, well, the thing to do with the plant was the surface area of the leaves. That was the key thing to do with it. The more and bigger leaves you have, the more surface area, and therefore the more surface area, you lost more water. A wee bit more detailed at the S level. You see there, leaf surface area, yes. So if you say, I think we'll just a quick look at essay question 11. It says, describe and explain how environmental factors affect the rate of transpiration in a plant. So it's looking at environmental factors. So let's just look at plant factors first of all. I think there's a couple of questions that do ask about plant factors. I'm just struggling to find. Yes, if you look at question 15b part 2, 15b part 2 is also then about 15b part 2. Question 15b part 2 is also about this. Yeah, so you'll see there. So the factors do with the plant are the surface area of the leaves. And you have to say, if you say surface area, you have to explain that. You have to say, the greater the surface area of the leaves, the more stomata you tend to have. Bigger leaves, more stomata. And therefore, the more stomata, the more openings. And you have to say it very clearly. More stomata means more openings for water to diffuse out. Okay. I suppose you could also argue bigger surface area of leaves, you get more cuticle. There's more top surface of the leaf as well, and to a certain extent then that increases water loss as well. But essentially it's about the fact there's more stomata and therefore more openings for water to right? Stomatal density. So you may have two very big leaves of the same size, but one may have low stomatal density and the other may have high stomatal density. That stomatal density simply means how many stomata per square centimetre. So higher stomatal density means more stomata and therefore more openings for water to diffuse out. Cuticle thickness, as we'll discover later, the thicker the cuticle. So these are all factors to do with the plant itself. Thickness of the cuticle. The thicker the cuticle, the less water escapes. And something else then is leaf hairs. Now leaf hairs get asked about fairly frequently. You'll see there, question 15b part 2. It says plants growing in dry environments often have leaf hairs extending from their lower epidermis. Where are these leaf hairs at? Well, I position the leaf hairs. I'm going to position them here on the lower surface. Imagine like a layer of hairs either side of the stomata. What's the purpose of the hairs? Well, if a plant wants, well, normally what happens when the water diffuses out through the stomata is that air moves the water away. So if you get water coming out here, so that's water vapor, it gets moved away by the air. And if you move the water away, then that ensures that this area here stays dry, that this area doesn't become humid. And if you can keep this area dry, then you've got an area inside that's very humid, a lot of water, high water potential in here, a lot of water, low water potential outside. Then the rate at which water moves out is quick. But what do the leaves do? Well, what the leaves do is they prevent air flowing easily over the bottom surface of the leaf, so the air can't get at the moisture that has accumulated outside the stomata. So the hairs, what the hairs do is they trap moist air, They keep the air that's moist outside the stomata. So the leaf hairs trap moist air close to the stomata. And that means this region out here becomes quite moist. And it gets so moist that it's almost as, the correct word is humid. It's as humid as inside, so there isn't much of a difference between the inside and the outside, and that reduces the diffusion of water out. So if you can maintain a high layer of moisture here, and that's what the leaves do, they trap the air and prevent the wind moving past, so they trap moist air and reduce the risk of water loss from the stomata. Now, plant factors then, environmental factors, and these are ones you're more familiar with at GCSE. They're dealt with in question 11B, which is the essay question, second part of the essay. Describe how environmental factors affect the rate of transpiration in a mesophytic. Well, they've told you there a mesophytic is a typical plant. We'll come to mesophytic plants in a moment or two. But essentially, mesophytic plants are the kind of plants you'd have here in our countryside. Sort of a medium sort of a plant that's not adapted to very dry conditions or very wet conditions, but can cope with our sort of climate, the mesophytic plant. And most of these factors you read about at GCSE, they were temperature, they were light intensity, humidity, and air movement, and so on. But you have to be very precise in answering this question. You have to be very clear about the effect of each of these. So you can't just say temperature increases the rate of transpiration. You need to say it clearly. You need to say higher temperature increases the rate of transpiration, and then you have to explain why. A higher temperature increases the rate of transpiration. You could argue because higher temperatures give the water molecules more kinetic energy, and the more kinetic energy, they diffuse faster. So higher temperatures makes diffusion happen faster, and diffusion happens faster, and the water molecules diffuse out more quickly out of the leaves. Also, higher temperatures makes the water molecules evaporate faster. So you can say water molecules have more kinetic energy. I've said that so they can evaporate faster. You can put a slash there and just write diffuse faster. So they can evaporate or diffuse faster. And that speeds up transpiration. Light intensity. Light intensity is an awkward one. You might say, well, if you shine more light on a leaf, how does that speed up transpiration? We're not talking about heat here. We're talking about a cold light, just a light, no heat. And how does light speed things up? Well, the thing about light is that the leaf responds to light by opening its stomata more. When leaves get more light, they open their stomata more so they can do more photosynthesis. So they can get in more CO2. But by opening their stomata more, then they let out more water. And so light intensity, more stomata are open, so more openings for diffusion water. Humidity. Well, humidity is the one that goes against the trend. If you increase the humidity outside a leaf, then you reduce the diffusion gradient, and you get a slower rate because essentially the amount of moisture outside the leaf becomes closer to the amount of moisture inside. So basically, higher humidity means a higher concentration of water molecules around the stomata. That decreases the diffusion gradient. Wind at GCSE understood if you blew wind past the leaf, that speeded up transpiration because the wind would blow away this water that accumulates there. Sometimes, by the way, some textbooks will mention that these the water molecules build up in what are called diffusion shells outside the leaf. So you can say that the wind blows away the accumulated water molecules or the diffusion shells of water molecules outside the leaf, and therefore that increases the difference between the diffusion gradient, and therefore that speeds up transpiration. So notice in answers to these questions, you have to use those terms diffusion gradient quite a lot in relation to these factors. Soil water availability. Less soil water available, and the stomata may close. Essentially, if you don't have water in the soil, you don't have water to move up through the plant, and therefore you don't get so much water being lost from the leaves. And another thing that's maybe not well known is that when a plant is stressed in this way, when a plant has very little moisture in the soil, it actually closes its stomata. It shuts its stomata, and therefore that greatly reduces the rate of transpiration. So moving on then, top page 10 there, how does transpiration of a plant vary during daytime and nighttime? And transpiration is faster during the daytime due to the high light intensity. So more stomata are open, so you get more transpiration happening during the daytime. And that part of the top page 10 relates to a question 1B part 2. It's not a very relevant question. I'll leave you to look at it yourself. Plant adaptations to climate. Well, this is what I was referring to earlier. You can categorize plants into mesophytic plants, which are everyday plant that you find around us in the countryside here, which cope with a sort of medium sort of climate in terms of moisture. a fairly humid climate that we would have with not very dry conditions and not very wet conditions, although you might argue we have a lot of wet conditions. But the plants we around are mostly referred to as mesophytes. But then you have plants which are designed to cope better with very dry conditions, or conditions where they're stressed with regards to water, where they don't get sufficient water or they're losing too much water. And these plants are referred to as xerophytes. And we immediately mind springs to the idea of like a cactus in a desert, cactus plants. And then the other end of the spectrum, then you have hydrophytes. And these would be plants that are well adapted to essentially living in water or very wet conditions. So they're exposed to very wet conditions. And these xerophytes and hydrophytes then have specific adaptations that allow them to survive in their extreme climates. And so I tend to get asked more about xerophytes. And the classic xerophyte is that you get shown in pictures is something called marram grass. Marram grass. Marram grass is the grass that you get down at the seaside, on the sand hills, on the sand dunes up from the beach. Those plants live on the sand there. And you might say, well, hang on, those plants are not exposed to very dry conditions or very hot, dry conditions, but they are still stressed for water, from the point of view that the sand that they grow in is very poor at retaining water. So there's very little water in the sand. And from the other perspective, the part of the plant above ground that's out in the air is constantly exposed to wind. So it always blows at the seaside. It's always blowing. There's always constant wind there. So the leaves are constantly being dried out by very dry by windy conditions, and the soil has no water in it. So essentially, those plants, those marram grass plants on the sand dunes at Port Stewart, Port Richmond, and so on, are xerophytes. They have adaptations to cope with these very dry conditions. And questions there, if you want to make a note just of the questions that relate to this, question 11a, question 12, 13, and 15. So 11a, 12, 13, and 15. So I'm looking first of all at question 11a, question 8, question 10. Yeah, so question 11 is the question 11, part A is the essay, and basically tells us about a xerophyte found in sand dunes. And then it says two leaf adaptations found in the xerophyte. And then it tells us also then about a hydrophyte then, and asks for one leaf adaptation. So two adaptations in the xerophyte. So the typical xerophyte then is marram grass, and you see there on my diagram on page 11 that cross section of the xerophyte and xerophytic leaf. So essentially, what is it? Well, if you imagine that a normal leaf, tall flat leaf, what in marram grass, what happens is the leaf is folded around like that. Folded around. And now this is the bottom of the leaf. This has got the stomata down here, and the stomata are effectively end up inside. So the leaf's kind of folded around into like a tube, and the stomata are all, so that's the bottom surface of the leaf inside. All the stomata are in here. On the outside here, you have the top surface of the leaf, which is all waxy cuticle. So waxy cuticle out here, bottom of the leaf in here with lots of stomata. Okay, and this is the first. So the fact that the leaf is rolled up, what's the significance of that? Well, reduced surface. See the surface area of the bottom of the leaf, that's reduced, it's exposed to air, and what happens is that as moisture comes out of the stomata inside here, stomata are all lining the inside, in the moisture, instead of blowing away quickly, it's retained inside the leaf, and that reduces the water potential gradient. So the fact that the leaf is folded up means that the stomata are on the inside the tube, as it were. The leaf's like a tube shape, stomata on there, reduced stomata gradient. And around those stomata then you've got lots of hairs. And as we said before, hairs trap moisture close to the bottom of the leaf, and that reduces water escaping, that reduces transpiration rate. In fact also the stomata, instead of the stomata, so in this particular question the stomata are out at the surface. If I change this a bit and made the stomata so that they were back from the surface a bit, so instead of the stomata being there, if I sort of bent the leaf back like that, and put the stomata up here. So that's one stomata, and then the next stomata is there. sorry, the next one, that's sorry, that's one. So there's the stomata now. Instead of them being out at the very surface, the stomata are back here. So there's like a kind of a cavity. There's kind of like a space outside the stomata, like a kind of a, so the stomata are like at the back of a cave, as it were. And then this is referred to as sunken stomata. And so what happens is that when water does diffuse out here, it kind of gets trapped in this hollow or cavity just outside the stomata, and that builds up. Water then is built up or is trapped in the depressions or cavities close to the stomata, and that reduces the water potential gradient again. Now, those are the main features. Sorry. Other feature is that the outside of the leaf has got a thicker waxy cuticle. So thicker waxy cuticle, and that reduces cuticular transpiration. So there's 4 key features of that plant. But in general, there are other features of plants that are xerophytes. And one of the other features is that they can have very deep roots. Having very deep roots means that they can extend, the roots can extend much deeper down into the soil to find water in a situation where there isn't much water. The other one is having succulent tissue. Succulent means juicy. And it comes to mind a cactus. If you look at a cactus looks more like a cucumber in shape. It's like a big juicy stem. But actually, that shape is referred to as having succulent tissue. Succulent tissue simply means lots of cells that can store water inside the plant, and so the water is stored and kept inside the plant. So that's xerophytes, and I would refer you then to all those questions on xerophytes. That's questions 11a, 12, 13, 15. Just have a quick look at 12, question 12. Which is like this. Yeah, 12 is a very old essay which asks you to compare the 3 different kinds of plants, which is a nice, simplistic, very simplistic kind of essay, and it's not too difficult to do. Question 13 is a more recent question, and it's showing you 3 leaves. You can see the bottom one there, the bottom of the 3 drawings. That's your marron grass again. So that's C is the xerophytic leaf, and give 2 adaptations, and how that's a very straightforward question. So the top 2 drawings are not important here, it's the bottom one. And then question 14, I think I also said question 15 is also about xerophytes. And let's see what question 15 is about. Yeah, well, essentially it's about stomatal density. Plants with different amounts of stomata, calculated stomatal density. Plants that grow in a very dry environment tend to have lower stomatal densities. Okay, moving on to hydrophytes. Hydrophytes then are plants that are adapted to grow in very wet or damp environments. Examples would be, and you may not know about these, but rice plants are an example. Rice plants grow in Asia, India, and the Asian countries. And if you ever see the growing of rice is a tall, a thin plant like a grass plant, and it's planted in fields that are full of water. So the fields are actually about that deep of water, and the plants are put into them, and that's their natural growing environment. So rice plants are hydrophytes. We have a natural hydrophyte in this country. It's not well known, but rushes, if you're familiar with with rushes, which are tall spiky fellows that you see there in farmers' fields in very wet ground, they are adapted to very wet conditions. So an example given here is that water lily. Water lilies are flat plants that lie on top of ponds, grow on top of ponds, and basically their main adaptations is they've got lots of air spaces inside the leaf, and that is to keep them floating so they don't sink in the water, and by floating they stay close to the surface. And close to the surface they get more light and more carbon dioxide for photosynthesis. The other adaptation is that they have their stomata on the top surface of the plant. This is relatively common. We're told the stomata are always on the lower surface of the leaf, but in this case stomata are on the top surface. Being on the top surface means they're in contact with the air. If they were on the bottom surface, then they'd be in contact with water, and water would flood in through the stomata, and the stomata couldn't get any carbon dioxide that way. So there's a couple of adaptations of hydrophytes. Now, uh, the last item here is translocation. So it would appear that most of this topic has been spent talking about xylem vessels and flow through the xylem vessels and loss of water and the ions, the water and ions moving up through the plant and through the leaf and so on and so forth, and the cohesion tension theory. And now we're only going to get a wee mention for the other kinds of pipes that are inside plants, and that's floam vessels. And floam vessels are about transporting the products of photosynthesis around the plant. Remember, photosynthesis happens in the leaves and you get glucose made. In photosynthesis, you move back to GCSE, you had 6 CO2s plus 6 H2Os give glucose, C6H12O6 plus oxygen. Well, that glucose that's made in the leaves is used in the leaf for lots of things, used for respiration, stored as starch, etc. But leaves can also export their glucose to other parts of the plant. In other words, this food that's made inside the leaf can be sent to other parts of the plant, and it's sent in the form of a different carbohydrate molecule, sucrose, S-U-C-R-O-S-E. And you remember back from your molecule section that sucrose was a disaccharide molecule made from glucose and fructose joined together. So whilst the leaf makes glucose in photosynthesis, if it wants to transport some of that food to some other part of the plant, it transports it in a different form called sucrose. And this sucrose moves through these pipes called floam vessels, and it's essentially dissolved in water. So I imagine moving through the floam a sort of weak sugary solution, because sucrose is table sugar, isn't it? A weak sugary solution moving through the floam vessels. And this movement of this weak sugary solution through the floam vessels is referred to as translocation. Translocation, moving the sucrose around the plant. And these, I'm not going to do any drawings on the board here. The best diagrams are here in my notes. As good a diagram as I'm going to be able to do anyway. And it basically shows you these xylem vessels are a bit different from floam vessels. First of all, they don't have any lignin, any thickening in their walls, any lignin. But they have these at the end of each floam vessel. At the end of these, the floam vessel is made up of things called sieve tube elements, which are like short sections of pipes that are joined end to end. And each one of these is something called a sieve tube element. Sorry, each one of these got something called a sieve plit at the end, and these sieve plits are like strainers or colanders. They've got lots of holes in them. Sieve tube elements positioned end to end. Sieve plits at the end. They porcelain material flow through. I'm referring you here to the two questions, question 14 and question 9. Just have a look at question 9 first of all. Question 9. Sorry, I'm going to refer you to another question. Let's have a look at question 14. Question 14, question 9. There was one other question that I want to look at first of all. I'm just going to tell you what it is. See if I can find it. Yeah, it's question 7. It's a good question to start with. And it says, xylem and floam are both found. Describe the distribution of xylem and floam tissues on the stem. And the answer to that is they're found in vascular bundles, which are positioned around the periphery of the stem, xylem on the inside, floam on the outside. In floam sieve tubes, the main cell types in floam tissue, two major features of floam sieve tubes. Floam sieve tubes have they have sieve plits at the end of each tube, yeah, sieve plits. And secondly, floam sieve tubes are associated with another cell. There's another cell alongside them called companion cells. So that would be the two answers to that. Part B, floam tubes are involved in the transport of organic molecules, while xylem vessels are involved in the transport of water and ions. Two differences in the way in which materials are transported in floam xylem vessels. Well, here's a very important point. Xylem vessels only carry water and ions up the plant, always from the root. This is xylem now, carrying water with ions, always upwards, always up to the leaves. Whereas floam vessels can be carrying sucrose down the plant, and down the stem, or up the stem. So in other words, movement in the floam vessels is both up and down the plant, two-way flow up and down the plant, whereas xylem's only upwards. Okay, I'll come back to that point in a minute or two. And yeah, the second point there, part B, second, is that I'll tell you the answer now and I'll come back to it. The movement of water in xylem vessels involves the cohesion tension theory, whereas the movement of sucrose in Floam vessels involves active transport and energy expenditure. So that's the answer to that. We'll come back to that in a slide or two. Name one organ of the plant which will receive organic molecules. So the organic molecules that's mentioned here, of course, is sucrose. And in other words, where might a leaf be transferring its, where might a leaf be moving some of its sucrose to get rid of that? Basically, the answer to this is it might be moving it to another leaf, to a younger leaf, for example. And so if you have, just do a quick plant, this is the stem of the plant, and here, let's say there's a, up here there's a leaf. Okay, so some of the sucrose that's down here, we have to say the roots of the plant. Okay, that leaf there might be sending some of its sucrose to other parts of the plant, and as I said, it can both go up or down. So some of it may go up, some of it may go down. And you might ask, where's that sucrose going to? Well, the answer is it could be going up, say, to another leaf somewhere up the top of the plant, a younger leaf that isn't maybe making as much sucrose. So it's maybe going to that leaf. But it may also be going up to the top of the plant, where at the top of the plant you may have berries or fruits or flowers or something. So up here you might have the plant might be making berries or fruits, to say that's a fruit or a berry or something that's been made up the top of the plant. So the sucrose may be going up there to that berry. Okay, or maybe going to a flower. There may be a flower up at the top of the plant. So say there's a flower up here. That's the flower. The flower may contain nectar. Remember, nectar was like a sweet material, a sugary material that attracts insects. Well, that's what the sucrose might be going up there. So it could be going to other leaves. It could be going up to flowers or buds or fruits. Well, if you think about what does an apple tree do? An apple tree has leaves that make all the food, the glucose, turn into sucrose. Then the sucrose is sent to the apples. And the apples then are like the berries here. So it could be a different, depending on the plant, it could be berries or fruits or whatever. That's where the sucrose is making its way to. Why might the sucrose be going down the plant? Well, many plants have down underground. Many plants have what you might describe as storage organs under the ground. So, for example, a potato plant sends its sucrose down underground to be stored in these things we call potatoes, which are simply like swellings on the root of the plant, where the sucrose is transferred down to there and then turned into starch. So your potatoes are really storage organs for the plant containing starch. So that's where the sucrose could be going up the plant or down the plant. Now, just in relation to that, there's a very, very good question on that. It's question 9. And question 9 really bamboozles students because they hadn't seen the like of it. I deal with it on the very back page here of the booklet. Question 9 shows a plant. And you'll see it relates to, on page 14 in the notes, at the bottom of the page, I've said translocation is two-way. In other words, translocation, movement of sucrose can be both up and down the plant. And that's what this question is about. It showed a plant, and it showed one leaf up at the top of the plant, and it said that you had CO2 been added. So that CO2 would be needed for the leaf to do photosynthesis. But it was odd CO2, it was C with a 14. That's radioactive carbon dioxide. Carbon 14 is radioactive carbon dioxide. And what they were doing in this experiment was they were supplying, instead of giving the leaf normal CO2, they were giving it radioactive CO2. And the idea would be that this C14 would be used by the leaf to make glucose. And then the glucose would be used to make sucrose. And then the sucrose could make its way out of that leaf and it could go around the plant. And basically what they were doing is they were following the movement of the sucrose around the plant using the radioactivity that had been locked into the glucose. And they're able to measure the amount of the amount of the carbon in the form of carbon-14 that was in sucrose that was traveling around different parts of the plant. And you can see from the figures that most of the sucrose ended up was still in the same stem, the leaf where it was added. But there's a lot of it down at the bottom, and a lot of it in the middle, and there's some here and some at the top and some at the bottom. So it says name the tissue in the stem where most of the radioactivity would be detected. In other words, where would you expect to find the sucrose? And the answer is phloem. And then name the organic substance, that would be sucrose. And then part B was the bit that really bamboozled students because they couldn't really get their head around this. But basically the answer to this was describe and explain the distribution of radioactivity. Well, describe the distribution. That's just describe. And you would say most of the radioactivity is in the stem of the leaf, or the part of it close to the leaf, there 10. But you would say there's quite a lot of it both up and down the plant. Quite a lot down at the bottom, quite a lot up at the top. That's the describe. Then your explanation would be that translocation of sucrose is both up and down the stem. So you would expect some of the sucrose that's made in that leaf or produced in that leaf to go down the stem and some to go up the stem. And then you had to explain why it would go down the stem. You could say it could go down the stem down to the roots, to storage organs to be stored, or it could go up the stem to fruits or buds or flowers up the top of the plant. So essentially that was that. That's a fairly random question, unlikely to be asked again. The other part of that question, I'm just going to come to a stop here soon. The other part of that question relates to, the other part of that topic is dealt with in question 14, I think, which is a very recent question about phloem, and it kind of shows a picture. It shows a Xylem vessel, and then it shows a subplate, that's a Floam vessel, and then it shows these cells that are beside the Floam vessels. So beside the pipes are a cell called a companion cell. And you'll see in my diagram what I say about companion cells. I said these cells are connected to the subtubes by Plasmodesmata. So these companion cells are found to have a lot of mitochondria. So you see in the picture there, those are supposed to be mitochondria. These companion cells have lots of mitochondria. When you can find lots of mitochondria in cells in biology, it suggests a lot of respiration, and that always suggests a lot of active transport. And that in effect is what's happening. These companion cells are very active. They're very metabolically active, and they're producing a lot of ATP. They have lots of mitochondria, and if you go to the top page 14, I kind of explain why. I say that these companion cells are involved in the movement of sucrose by translocation. Companion cells have many mitochondria to provide ATP, and what they do is that companion cells provide ATP so that lots of sucrose that's made in the leaf can be pumped into the companion cells by active transport, can be moved in by active transport, such that the companion cells become so full of sucrose that the sucrose actually leaks out of the companion cells into the subtube elements, which are alongside, and it moves into the subtube elements by plasmodesmata, through plasmodesmata strands. So essentially what we're seeing here is the movement of sucrose is an active process. It involves these companion cells and all this active transport and ATP and mitochondria and so on. What's the evidence? How do we know this? Well, the evidence is that in experiments, if you do anything to the phloem tissue that stops transpiration, that stops respiration, then you can stop the movement of sugars by translocation. And so you can do various things. You can starve the phloem tissue of oxygen. That means the companion cells don't get oxygen due respiration, and so you don't get any movement. You don't get any ATP made, and therefore you don't get any movement of sucrose. Or you can do things to the phloem tissue. You can use a chemical called cyanide that stops respiration. And once again, if you stop respiration, you stop translocation. So that's the evidence that translocation is a two-way flow. Just looking at question 14 is quite difficult. 14a, part 1, state the function of the companion cells. You could say the companion cells provide energy for the movement of sucrose into the subtube elements. State two ways in which the process of transport in xylem differs from that in phloem. Transport in xylem is both in xylem is always up from the root, up the plant, whereas transport in phloem can be up the plant or down the plant. And the second thing is transport in xylem involves something called the cohesion tension ideas, whereas transporting the phloem involves a metabolic activity, involves the release of energy. Part B is a fairly random bit, and yep, it's really essentially about reading figures and so on, so you can look through that yourself. Okay, that's plant transfer, quite a difficult topic, but very relevant to topics you'll do at A2, so it's useful to understand it quite well, because at A2 you'll be doing topics to do with photosynthesis and respiration, and you'll be doing ecology topics which involve you understanding a bit about plants and photosynthesis and transpiration and so on and so forth. So there you are. Thank you very much, and all the best.